Hi, this is Phil Newman. I'm the editor-in-chief of Longevity.Technology, and I'm delighted to say that I'm joined today by Dr. Joshua Hare, chairman of the NASDAQ-listed company Longeveron. Hey, Dr. Hare.
Hi, how are you? Pleasure to be with you.
Wonderful. And we know that Longeveron is a clinical stage biotech company doing some excellent work in cellular therapies for chronic diseases. So, Dr. Hare, could you maybe just give us a little background on yourself and the work that you're doing at Longeveron?
Yes, my name's Joshua Hare. I'm a cardiologist. I work in the city of Miami at the University of Miami. We founded Longeveron 8 years ago as a spin out from the University of Miami. The company took a license from the University of Miami to its Lomecel-B technology. And we have, for the last 8 years, been doing clinical trials focused on aging-related conditions. So we have a strong emphasis on aging frailty, and Alzheimer's disease.
Wonderful. And there's a lot of excellent work that we're going to be talking about today, I'm sure. But maybe if I could just go back a little bit about the decline in mesenchymal stem cells and how they contribute to aging, and how Longeveron is working with this particular area.
Yeah, that's a great question. One of the reasons we were drawn to the field of aging is because of the observation that there is a decline or a deficiency in adult stem cell reservoirs in the human body. And in fact, as we've started to understand aging as a biological process, one of the key pillars of aging is a deficiency in adult stem cells in the body. So it made sense to us to test the idea, if stem cell depletion is contributing to aging, can we replete those cells with an external infusion of the cells, and can that have a favorable impact on aging phenomenon such as frailty or cognitive decline?
Which, of course, are two of the big areas where aging science needs to show its effects. And of course, the fact that you are now moving into your phase 3 trial is obviously very exciting for the industry and likewise for your company. So, Dr. Hare, what makes Longeveron's up-and-coming phase 3 trial so pivotal?
We are in discussions with the Food and Drug Administration in the US and the PMDA in Japan about our future trials. We haven't yet received regulatory approval to do the phase 3, but we have completed a phase 2B trial in the United States. And it's armed with that we will be having discussions about the next step and the next pivotal trial with FDA. We're very excited about the results because we used 6-minute walk distance as the endpoint for our trial, the primary endpoint. 6-minute walk distance is a very nice integrated measure of functional capacity in people, and it declines with age and declines faster with people with aging frailty. So we enrolled people in the study who already had a depressed 6-minute walk distance. And the results of the study were that there was a dose response increase in 6-minute walk distance in older individuals with frailty. So it's with that in hand that we go to the FDA for discussions about the next pivotal trial that we plan to conduct.
That sounds excellent. Now, I guess the fact that you're moving into phase 3 begs the question about commercialization and the funding routes associated with funding a phase 3 trial. So what is the commercialization route that you have for Lomecel-B and some of your other cell-based product candidates? I mean, are you looking to partner with Big Pharma? Or are you looking to go all the way yourselves? Just interested to know what the plans are there.
We're open to either pathway. We are a publicly traded company, so we have access to the capital markets. And also, however, we're very cognizant of the challenges in commercialization and having sales forces and getting reimbursement approval. So we would be totally open to a partnership or a self-funded effort, depending on the circumstances that confront us.
Understood. And of course, the Lomecel-B product has been on your website is talked about as being an off-the-shelf product. So interested to know how you would define that as being an off-the-shelf product.
Right. So this is one of the very exciting features in the cell therapy space. Cell therapy is emerging as a whole new paradigm in medicine. The idea of treating diseases with cells that are very targeted and perhaps might have a greater safety profile than pharmaceuticals. The issue of off the shelf is an immunologic issue, because if you're taking a cell population, you want to make sure that there isn't no immune rejection, or that you don't have to use immunosuppressive therapy. Now, the whole issue with mesenchymal stromal cells, also known as medicinal signaling cells, is that they are safe to administer as an allograft. They lack the class 2 major histocompatibility complex antigens, and they have some other features that make them safe to give as an allograft. So it is that property that makes them off the shelf. So we can make these cells in large batches, have them cryopreserved, and then ready to give when a patient needs them. It's a major advantage, and I think it allows a scale up of cell therapy tremendously in a way that auto therapy just can't accomplish, because in auto therapy, everybody's got to be their own donor.
Yeah, and I guess that's one of the things that's very interesting from an investment perspective. Just sticking with frailty just for a while, I'd like to go on to Alzheimer's in a moment, but frailty is often misunderstood, and it's very difficult to identify systematically. So, I mean, can you tell me a little bit more about your approach to your multi-trial approach to frailty, and how you're going about constructing those trials?
Yes, so you hit the nail on the head about frailty. There are a lot of misconceptions, and many people, even physicians, consider frailty an inevitable consequence of aging. Geriatricians and basic scientists who study it know that that is not the case. Frailty is a biologically driven process that occurs in some, but not all people. Certainly age is a key factor, and the older you get, the greater risk you have for frailty. But there are plenty of examples of people who are the same age and one is frail, the other isn't. This goes back to the question you asked me about cell depletion, because as studies have been done on what the biological basis of frailty is, we know that stem cell depletion is one of the factors. Another factor is inflammaging, or an inflammation, a low-grade inflammation. This is also relevant to Alzheimer's when we talk about it. So there are classification schemes, there are clinical classification schemes. There are 2 or 3 of them that are very relevant. And there's a growing awareness in the medical community that you can diagnose frailty, that there are scales and scores that can be applied, and that we are now at the very first stages of trying to treat it. A very famous physician from last century, Sir William Osler, stated that the first step in treatment is diagnosis. And we are now much more comfortable about diagnosing frailty. And that is a critical first step to then starting to do treatments. And in our trials, we defined it using a clinical scoring scale, but we also added specific features of 6-minute walk test. We only took people whose 6-minute walk test was already depressed down from what a normal healthy adult would be. And so we used that as an inclusion criteria in our trial.
And in relation to, of course, we're all experienced within our own families, whether a relative has perhaps clinical decline mentally, or they suffer from frailty, or unfortunately, sometimes they'll suffer with both. Sounds very interesting in terms of what Lomecel-B has in terms of its multiple methods of action, and could obviously simultaneously address Alzheimer's. So really interested to know what your preclinical studies are showing in the area of cognitive decline and Alzheimer's.
Yes, most of our preclinical work is done in heart failure models, but we have done, there is a lot of work on Alzheimer's between our laboratory and many others. And there are animal models of Alzheimer's. Now, the key issue and the hypothesis arises of why would you use cell therapy to treat Alzheimer's? It's not necessarily to try to replace missing neurons, but it's more to treat the inflammaging or the neuroinflammation that accompanies cognitive disorders. And so that's one of the things that's not addressed by other approaches, which are based only on removing amyloid or tau proteins from the brain. So we know that there's a lot of neuroinflammation. And this applies not only to cognitive decline, but also emotional regulatory disorders like depression. So the brain does get inflamed. The cell-based therapy is very effective at restoring immunological milieu back to normal. And we believe that that is one of the key mechanisms of action by which cell therapy can address frailty and cognitive decline. It's through immune modulation. So another key mechanism of action of the cell-based therapy is its effects on microvascular endothelium. The microvasculature is a network of vessels that courses through every organ in our body. The endothelial dysfunction of those vessels is also something that increases with age and is increased with diseases like hypertension or hypercholesterolemia. We think that we think that microvascular endothelial dysfunction plays a key role in cognitive decline and in skeletal muscle disorders like frailty. So another key mechanism that we're looking at, in addition to inflammation with Lomecel-B treatment, is improvement in microvascular endothelium. And we're measuring biomarkers of that. And we're also trying to measure endothelial dysfunction directly with newer techniques.
Interesting. And just, if I may just touch on biomarkers, because when you're looking at frailty, you're looking at cognitive decline, you're also looking at microvasculature. There's an interesting panel that you're looking at in terms of those biomarkers. So just before we finish today, I'd love to know a little bit more about what you're doing in terms of benchmarking what you're doing against your own science with recognized biomarkers.
Yes. So we're actually going beyond recognized biomarkers, and we have a whole biomarker discovery program. This is something that regulatory authorities are very interested in, and in medical societies, because it's always nice to have something you can measure in the bloodstream that correlates with the bioactivity of the drug. Our work in that area is premature, but we have done and measured panels of both inflammatory and vascular biomarkers in all of our patients, both those with cognitive decline and those with skeletal muscle sarcopenia and frailty. That work is ongoing, but I could tell you we've measured panels of over 20 biomarkers, and we're analyzing the data right now. I can tell you preliminarily that we're very excited by what we're seeing. That's preliminary, and we hope to be able to announce more robust results in the future.
It all sounds wonderful. So, Dr. Hare, just to finish off, what's an average day like for you now? You've got quite a lot on your plate.
I'm very, very busy. I love it. It's a great career path. I'm still clinically active, so I have patients. I still teach and do research at the university. And then my operative role at Longeveron is as the chief science officer. So I engage with the scientific team and get to participate as an advisor in the types of studies that are being planned. So it's very exciting. And I think this field of cell-based therapy is just at its infancy. I think we're going to see amazing advances in the next few decades, and to be able to be involved as a basic investigator in advancing this field is really very exciting.
Well, wonderful. It sounds like great work, Dr. Hare, and we look forward to learning how you get on in your phase 3 trials.
Thank you so much for having me on the program.
Wonderful to see you. Thanks very much, Dr. Hare.